So let's continue from where we stopped last time. We had been discussing about the RNA and its types. Okay, what is the full form of RNA?
Ribonucleic acid.
Ribose nucleic acid.
Very good. It is ribose nucleic acid, and we have discussed about the differences between DNA and RNA. Correct. So the one major point is that DNA is a double-stranded molecule, while RNA is a single-stranded molecule. Okay. And then in the other major difference is in one of the nitrogenous bases. DNA has got a thymine, which is replaced by what in RNA?
Uracil.
Correct. It is represented by U. And by now, I think everybody should be very well aware about something called complementary base pairing. All of you are aware? Adenine pairs with guanine. Okay. And thymine pairs with cytosine.
Cytosine.
Correct. Okay. So that's called complementary base pairing. So some practice questions based on that we have already done. Right. And all of you remember we had started with the structure of mRNA. You remember something called codon?
Yes.
Right? Yes, we had discussed about the word codon. Codon is nothing but a set of 3 nitrogenous bases on the mRNA. So that codon we read in which direction? What is the direction for codon?
5 point to 3 point direction.
Correct. It is 5' to 3' direction. Okay. So this is the mRNA that I showed you. 3 of them taken at a time. And we have also discussed why a codon consists of only 3 bases. Why not 1, why not 2, or why not more than 3? Correct. Totally, how many amino acids are there in the biological world?
20.
20 amino acids, right? So the important point, the correlation between a codon and an amino acid is that a single codon codes for 1 amino acid. That is a very important point you should remember. Okay, so that's about the codon. And then we also spoke about something called start codon, also called the initiation codon. Okay, and that is always AUG. Okay, similarly, there is something called stop or the termination codon. Okay, so this stop or termination is not one codon, there are 3 of them, and any given mRNA, it can be any one of these 3. Okay, so we can know it by UAA, UGA, UAG. So these are the 3 stop or termination codons. Okay, these 3 are also actually known as nonsense codons. Okay, इनको literally हम लोग nonsense codons बोलते हैं. Okay, why it is called nonsense? किसको बोलते हैं nonsense? Somebody who doesn't make sense, right? Friends के बीच में होता है ना कभी-कभी कुछ भी बोल दिया तो क्या बोलते हैं? What nonsense are you talking? So that time what you are going to revise? Stop codon. Okay, because we know very well is stop या termination codon. This 3 does not code for any amino acid. इसीलिए उसको nonsense codon बोलते हैं. All the other codons, I have shown you that table last time. Okay, totally considering all the codons possible, we have a set of 64 possible codons. Okay, out of the 64, these 3 are not coding for anything. So other 61 are sensible codons. These 3 are nonsense codons. All these points you have to remember. Okay, and codon is also called genetic code that we said. Every codon is coding for 1 amino acid. Okay, and we all know very well that ultimately why are we discussing about amino acids and all? Because any DNA is able to express its character by producing a protein. Okay, so a DNA expresses by producing protein. That means it has to manufacture a protein. And for protein, the raw material is amino acids. Getting it? So a DNA giving rise to a protein is possible only when there is a RNA present. DNA takes the help of the RNA. Okay? So we'll see that table once again. See this. This is the genetic code dictionary. All of you remember this? This is not given in your textbook, right? Yeah, but this is very important. The entire process is given in the textbook, but the dictionary nahi diya hua hai. So it is very difficult to understand that way. Okay, you don't have to by heart this dictionary at all. Okay, but just for reference. So इसमें क्या करना है? Codon is consisting of 3 letters. So first letter over here. Okay, then you have the second letter here, third letter here. So I take any one of the 4 letters as the first letter. For example, I take G. Okay, so second letter I take, for example, A. Third letter I would like to take U. Okay, so G, A, U. So where is it written? See this. Here, no? So this is one random codon. Okay, GAU. So this one is coding for which amino acid? It is written as ASP. So what is the meaning of that? ASP stands for aspartic acid. These are the 20 amino acids. Getting it? Easy to understand? I don't think it's very difficult. Anybody is having any query? Please ask. Everybody understood how to read the genetic code dictionary? Yes, you don't need to by heart anything of this. This is for your understanding. Okay, but you have to understand how to read it. Okay, so as you can see, this is an amino acid. For example, I take this one, LEU. LEU is written as leucine. Okay, so leucine, if I take as one amino acid, it is coded by 4 of these codons, you can see. So that means what? For one amino acid, there are 4 possible codons. Okay, but if you take a codon, say CUU. So CUU, as you can see, is coding only for leucine. Can you find CUU written anywhere else? Coding for any other amino acid?
No.
No. So you understood the meaning? That is the point. Okay, an amino acid can be coded by more than one codon. But any codon codes for only one amino acid. That's a very important point. Okay, now everybody look at the table and I will ask you verbally. I'll tell a codon, tell me it codes for which amino acid. Ready? Okay, so the codon I tell is say CGC. CGC codes for which one?
Arginine.
Arginine, correct. Okay, so you found it correctly, right? One more. Ready, everyone?
Yes.
Everybody should answer, okay? Tell me the codon AAA codes for which one? Triple A.
Lysine.
It is? Repeat again.
Asparagine.
No. AAA. See this one.
Lysine. Lysine.
Lysine. Okay. Most of the amino acids, if you see, their names end with INE. In. So you have to pronounce it as in. Serine, valine, proline, lysine. Okay. ऐसे बोलना है. Just a pronunciation. Okay. Okay. Now the reverse way. If I ask you any one codon for the amino acid threonine. threonine. Any one codon.
ACU.
ACG.
ACU, ACG. What about others? Ishika, what is your answer?
ACC.
ACC. Ishika, what's your answer? Threonine.
ACC. ACC.
ACC. Sundar,
ACU.
ACO. So everybody got it right. Like threonine is this one. Okay, good. Next one. Tell me the amino acid for UGA.
Stop codon.
Stop
Yes,
codon.
it's a stop
Stop
codon. You did not have to search in this, right? UAA, UAG, UGA. These three are stop codons. Okay. Now, can you spot the start codon or the initiation codon? And tell me it codes for which
AUG.
AUG.
AUG. So it is coding for which amino acid?
Methionine.
Very good. It is methionine. Okay, so this point also you have to remember that in every mRNA, the start codon is AUG, that is fixed. And that always codes for only one amino acid, that is methionine. Okay, so now we'll move ahead to one more question. Now you can see I'm giving you a sequence on the screen. Okay, all of you can see here at the top, there is a sequence I have given, a random mRNA sequence. Okay, so it starts from mRNA. The 5' end. Now there are codons written one after the other, ending at 3'. So what you have to do? क्या करना होगा बताओ? Looking at the sequence. Looking at the sequence, referring to this genetic code dictionary, you have to write the list of amino acids one after the other. It's a chain of amino acids, in fact. Last time I had mentioned 2 amino acids are bonded to each other by which bond? Two amino acids in a protein are bonded to each other by which bond? Remain on the screen, everyone. Keep the videos on throughout. Which bond is present between 2 amino acids?
Peptide.
It is peptide bond. P-E-P-T-I-D-E, peptide. Okay, remember that. So that means here in the sequence which I have given you, just look at the sequence and tell me how many codons are mentioned. How many codons can you see?
10.
10.
Everybody getting the same answer? 10 codons? Okay, so how many amino acids you must be getting?
10.
Are you sure? You will get 10 amino acids?
9 because last
9.
one is stop codon.
Exactly, yes. You will get 9 amino acids. So how many peptide bonds?
8.
Getting it, everyone? So if you have a given sequence of an mRNA, okay, if n is the number of codons, n minus 1 would be the number of amino acids, n minus 2 would be the number of peptide bonds. Got it? So can you make the sequence of amino acids for this? Looking at the table?
Yes.
Yes. Okay, I'll give you 2 minutes time. Write the sequence and see. Everyone. Again, all of your videos have went off. Keep the videos on, I say. In a classroom, we are always in front of each other, right? So this is a virtual classroom. You can write the symbols for the amino acids which are given, 3-letter symbols. Got it, everyone?
Done, sir.
Wait, I'll ask. Okay. What about others?
Done, sir.
Done? What about Sundar, Shivganga, Ishika, Harsha? Anybody who has not understood or is not able to do it, you can ask. Ishika, are you able to understand how to do it?
Yes, sir.
Good.
Done, sir.
Okay. Okay. So one by one, I will ask. You can tell me the answer. Okay. Okay. Abhirami, tell me what is the first amino acid?
Methionine.
Methionine. Very good. Diwali, next one.
Arginine.
It is CGA. Okay. Okay. That's right. Okay. Then what about Harsha? Next one.
Histidine.
Histidine. Very good. Sundar, GUU.
Valine.
Valine. Correct. Ishika, UUU. What is it? Yes, Ishika.
Proline.
Sorry.
Proline.
UUU. Is it proline? See here. PHE लिखा हुआ है. What is PHE? Read this.
Phenylene.
It is phenylalanine. Repeat. It is phenylalanine. बोलो.
phenylalanine,
Okay. Okay. Shivganga. UUG.
Leucine.
Leucine. Very good. Then कौन बचा है? Okay. Anybody? G. क्या है बताओ?
glycine,
Glycine. Correct. What is
lysine,
this? AAG.
lysine,
Lysine.
Very good. UAU.
triosine, triosine,
Triosine.
This one you are telling, right? It is tyrosine. Okay. Good. And last one.
stop codon,
Stop
So
codon.
you just write a stop. Okay. Okay. Good. So this what you have done. Okay. The codon was shown to you, or the mRNA was shown to you, and you have deciphered by creating a sequence of amino acids. This is called as amino acid sequencing. ये जो हमने किया अभी, this is called as amino acid sequencing. Okay. So same way, there is one more question. This you can solve later. I'll ask in Google Classroom. Everybody will post your answers over there. Same way you have to do it. Okay. You understood how to do it, everybody?
yes, sir.
Right? Shall we move to the next
Yes,
one now? Okay. I hope
Yes,
this is interesting for everyone.
Yes.
Yes, I
Yes,
was
sir.
expecting one question. If some of you would have asked, how do we know that this particular codon is coding for some particular amino acid? We are studying as if we are aware about it. ये हमें कैसे पता चला? Was it written under the microscope that this is the amino acid, this one codes for this one? How are we aware about it? Any idea? Somebody must have discovered it, right? Somebody has discovered this important point, that a particular codon codes for an amino acid, and this discovery was a very important discovery. Okay, and this entire discovery is called deciphering of the genetic code. It's like completely a coded language was encoded. A very important part. And we should be proud to know that this was done by an Indian scientist. Okay, he was a scientist from Punjab, Dr. Hargobind Khurana. He got a Nobel Prize for this. He was the one who is credited with the discovery of the entire genetic code. That's what we are referring to now. Did you understand? So remember the name of the person, Dr. Hargobind Khurana. Or simply you can say Dr. H.G. Khurana. Okay, he was given a Nobel Prize for this huge discovery. Okay? And this part of biology that we are learning, protein synthesis, functioning of the DNA, okay, mRNA structure, their functioning, this comes under a branch of biology which is known as molecular biology. Okay, and molecular biology is a very vast subject. You all are learning this from the very initial stages, but it's a very vast subject and many discoveries are done. Still, there are many things which we don't know, even after all these discoveries. Okay, so there are people, there are researchers working everywhere, and there are a lot of research institutes where people are engaged in all this. Getting it? So this becomes a very interesting aspect. Got it, everyone? So mRNA is clear. Okay. In the previous lecture, I also mentioned that the non-genetic RNA are of three types. Which are the three types I said?
mRNA, tRNA, and
mRNA,
rRNA.
tRNA,
Correct.
rRNA.
mRT. Very good. Here we spoke about mRNA. What does the M stand for?
Messenger.
Message.
Messenger, not message. It is messenger. Okay. What is the literal meaning of the word messenger? Messenger word का normal meaning क्या है? somebody who passes on a message, उसको messenger बोलते हैं, correct? So now whatever about mRNA which you have understood, do you agree with the fact that mRNA does the work of passing on some message, right? mRNA is carrying a message, right? It is having a sequence of codons, which is nothing but the message that these are the amino acids which have to be joined. And that's the reason we call it messenger RNA. Understood this? Okay. Okay. So let's move to the next type now. After mRNA, now the next one that we have is tRNA. Okay. Now tRNA, the T stands for transfer. T stands for transfer. Okay. So yeah. Now mRNA is the one which is carrying the message. Now the message has to be transferred, and that is done by the tRNA. Okay. Where is the mRNA produced? Now that's another important point we missed out. mRNA, where is it produced? Any idea? Okay, I'll write something on the screen. mRNA is produced from the DNA itself. Okay, that's again a big process. In fact, mRNA is produced from the DNA in the nucleus, and that process is called as transcription. The process of production of mRNA from the DNA is known as transcription. This important point you have to remember. Okay, then afterwards what happens? mRNA moves out of the nucleus into the cytoplasm and ribosomes. Okay, inside the nucleus, transcription takes place, mRNA is produced. That mRNA comes out of the nucleus, okay, in the cytoplasm, where what is present? The ribosomes are present. Ribosomes are present in the cytoplasm as well as attached to the endoplasmic reticulum. So mRNA will move towards that. We will see what happens after that later. For now, just understand this much. The mRNA produced inside the nucleus, coming out of the nucleus, going into the ribosomes and remaining there. Okay? So that was about the mRNA. Remember the word transcription also. Okay? Now we have to discuss about tRNA and where is it present, what is its role. Okay? Just do one thing, disconnect and join back again. The time will get over. So we'll start with tRNA.